Greetings, friends, fellow Earthlings, and relatively large living things. Welcome to Ask An Astrobiologist, the show that celebrates the science and celebrates the scientists involved in our quest to understand the nature of life. I'm your host, Dr. Graham, the cosmobiologist Lau, and we're brought to you by the NASA Astrobiology Program and SegaNet.org. As always, we want to thank all of you out there in the interwebs who share about our show, who interact with our guests during the live stream. We love highlighting the things that you do and the ways that you get involved in sharing in this journey of astrobiology with all of us. This month, we want to highlight Connor J. Ballard, who is a PhD student in astrobiology and geology at University College of London, for giving us a shout out on Twitter recently about our last month's episode. It's always fun to have all of you engage in any ways you can. I love engaging with you on any place in the social media realm out there, and there are certainly a lot of social media places out there to engage with. Today's episode is going to be pretty exciting. We're going down to the world of the small today, the very small. Our guest for this episode is Dr. Gary Trubl, and I'm so excited to finally have him on the show. I've been thinking about having him here for a while, but he just recently had a new paper published, and that made me think, well, it's time. We have to have him on the show. Dr. Trubl earned his bachelor's degree at the University of Arizona in environmental sciences, and then earned a master's degree in environmental science and health at the University of Nevada in Reno, while also working at the Desert Research Institute, where he was doing some research into determining the origin of N2O in Lake Vita brines in Antarctica. Dr. Trubl then went on to his PhD in microbiology at The Ohio State University, where he worked on characterizing viruses in Arctic peatlands along a permafrost thaw gradient to understand their roles in permafrost thaw and ecosystem carbon processing. All very cool research, of course. Dr. Trubl then went on to do his postdoc and is now a research staff scientist at the Lawrence Livermore National Laboratory. His ongoing research here, Gary's using multiple meta-omic approaches like metagenomics and metatranscriptomics and viromics in combination with stable isotopes and radioisotope probing to better understand the mechanisms that viruses use to control microbial physiology and to quantify nutrient exchanges in soils. So I'm so excited to have him join us. Gary, thanks so much for joining us here on Ask an Astrobiologist.
Hi, thanks for having me on.
Like I said, I've been, kind of wanting you on the show for a while. I've been super excited by the work you've done. You hosted a NASA Astrobiology Institute workshop some years back on this realm of astrovirology. And so today we're going to talk a lot about viruses, what they are, your research in viruses, and astrovirology. But before we get there, I always love for our guests to share kind of their pathway, their science origin story that has brought them to where they are. Would you mind laying out for us what the moment was for you where you realized this was going to be your career?
Yeah, that's tricky. I don't know if there was just a single moment or accumulation of many moments. When I went in to do my undergrad, I had no idea what I wanted to do. I tried veterinary science. I tried political science. I hopped around. Environmental science kind of spanned everything, and you could focus in different areas. So I kind of went with that, and that was enjoyable. I got involved in undergrad research lab, which really helped me go to graduate school. During my grad school, I started tinkering around. I realized I like cold environments. I like extreme stuff. Like, what are the limits of life? So I started working in Antarctic research, and I enjoyed that. But then I really wanted to know, what's all this new meta-omic stuff that's out there? There was barely any courses when I was an undergrad. So then I moved on to do a PhD using those tools. During my PhD, I was working on a permafrost thaw gradient. I was originally going to look at the bacteria in archaea, but then I came across Maya Breitbart's 2012 paper, Marine Viruses, Truth or Dare. And maybe if you're going to talk about one moment, that was the paper that just changed my life forever. It is captivating, it's engaging, it's well written, and it's even got these great cartoons that really show you why viruses are important. And before that, I had not taken a virology course, but I took a microbiology course. And, you learn about human viruses, HIV, herpes, kind of boring. I didn't really find them too exciting, right? I wanted to know about ecology, how they interact with everything around them. And I didn't really know this was possible. But then after reading Maya's paper, I was hooked.
Oh, very cool. And I feel like, our understanding of the biological realm around us, of course, it's evolved over time, but understanding viruses and their roles feels like a fairly new thing. compared to understanding microorganisms, other things like bacteria and archaea, or understanding larger creatures like plants, animals, and fungi. It's viruses are so small. It seems like it's just taken us a long time to really engage with our understanding of them.
Yeah, I think that's exactly right. I mean, if we go back 30, 40 years ago, what we're talking about for viruses was the same for microbes, right? We were just figuring out, oh, archaea is a thing, this bacteria, what's going on with that? And I think in 20, 30 years from now, it's going to be the next smallest thing, right? So it's great how our tools are progressing and allowing us to see even smaller things each year.
Absolutely, and it's science has unlocked scales of time and space for us that we did not evolve to think about, quite honestly. And then our knowledge, in astrobiology, our knowledge is evolving of what life even really is or how to characterize life. I did put up a poll on my own Twitter account last night, just asking the question that I'm sure you get all the time: Are viruses alive? And of the answers of yes, no, maybe, or what's a virus, about 60%, almost 60% of the respondents said maybe a little bit of both, maybe alive and not alive. I wonder if you can give us a little virology 101 of what our current knowledge is of viruses and how they interact with the environment.
Of course. So I appreciate you putting it out there, and I appreciate you even giving the option of more than a yes or no. Because as we're seeing with life, everything's a spectrum. There's no black, there's no white. It's all a spectrum for everything that comes to do with biology. So I do get that question a lot, and it's somewhat frustrating. And the reason why it's frustrating is we still don't have a definition of what life is. We have working definition for something we may see, but if you actually look at the support for it, it continuously changes. If you think about, all right, what are the characteristics of life? Well, they have to grow. Crystals grow. They're not life, right? So what exactly is needed? And currently we have the tree of life, which is based on, a set of marker genes. And currently viruses don't have, or we haven't found a virus, I wouldn't say they do though, to have this marker gene. So they're not included in the tree of life. But to say viruses are alive or not is it's interesting, because if we went to another planet and we found a virus, would we really argue that there's not life there? So if we talk about the things we know about viruses, they need a host. So if we found a virus, we know there has to be a host nearby. Viruses themselves don't have metabolism. So maybe if we look at the virus particle itself, so we call it a virion, it's when you have the virus shape, and then you also have nucleic acid, which can be DNA or RNA. Maybe it's not active in that state, right? There's not generation of ATP. But sure enough, when it infects a host, it takes advantage of the host. It reprograms the host. It uses the host's metabolic machinery. And it does what the virus wants. So at that point, it must be the virus, right? The virus is now alive. So I would definitely argue a little bit of both. And just to say a hard no or a hard yes is hard to say.
That's awesome. Yeah. And I will say some of the commenters on that post, they brought up exactly things like that. We had a conversation about information theory and whether we should be characterizing life by how it stores and transfers information, which viruses most certainly do. And then one person even mentioned, like that, the viruses might not be alive. And I did pose that question: what happens if we find a viral particle on Mars? Even though it doesn't show us the cell that it could be infecting, I think a lot of us would argue that counts as a life detection. And so, yeah, there's a lot for us to explore there and to learn about viruses. Now, I think, you mentioned earlier kind of how, a lot of our thinking about viruses, especially, with the recent coronavirus pandemic that we've all suffered through together. We all know about things like, HIV and some of these other viruses that infect humans and cause, everything, colds, all kinds of stuff. But we don't think as often about the other kinds of viruses that are out there and how they are involved in the environment. And that's been more of your research. I wonder if you can give us kind of your vision of viruses as they are involved in the environment in an ecological kind of sense here on Earth.
Yeah, and that's exactly what got me excited about viruses. It's not that I don't care about humans. It's just that we're looking at the minority of viruses because we're human-centric, right? We care about things that are relevant to us, and that makes sense. But if we look at what the majority of viruses are out there, it's such a diverse term And what I've spent most of my time studying are actually bacteriophage, which are viruses that kill bacteria. And there's more bacteriophage than there are of any other type of virus out there, right? You have bacteriophage, you have viruses all over you. And I like to say viruses as a loose term because there's so many other viruses. There's microviruses, which are viruses that infect fungi, archaeoviruses, anything out there gets infected by a virus, including viruses themselves. So when we're thinking about, oh, viruses is bad, viruses is good, that's not how it is. We have to think about the ecology. We have to think about the ecosystem. What are they all doing? They're all playing a component in the ecosystem, right? And so I like to think of things at a systems level. And I started going with soils because soils are arguably the most diverse system out there. And they are really tough to study. And I love it, though, because we interact with soil. We grow up with soil. It's all around us. There's so many ecosystem processes that soils gives us. And in soils, it's not just one thing is doing this. It's this affecting this, affecting this. There's a cascade of effects. And we need to know how they all interact as an ecosystem as a whole. And this is part of the reason why I use the metagenomics, the metatranscriptomics. And to give everyone a background, this is where instead of looking at an ecosystem, we're not pulling out one bug and saying, hey, what are you doing? We are extracting nucleic acid, so DNA from metagenome or RNA from metatranscriptome of the environment. So we take one sample. We have some soil here. We extract all of the DNA out. When we extract that DNA, we sequence it so we can put it on the computer and see what each individual does. And we have to bring them back and reconstruct their genomes. And then we figure out how they're interacting. And we can't just look at DNA alone because DNA tells us what is the baseline, what's the potential, what do they encode, what can they possibly do? Not that they do it, though. The metatranscriptomes is when we start looking at RNA. So we have to go from DNA to RNA. So RNA shows, hey, this is metabolism that's happening, right? But we also kind of preempt things, right? So a great example would be in soils when it's going to rain, right when the rain is about to happen. You smell that, especially if you're in a desert environment. I grew up in Arizona. I love the smell of the desert before, during, and after a rain. That geosmin smells so good, that dirt smell. So the microbes get ready, right? They start chugging out mRNA, which is the messenger RNA. They're getting ready if it's going to rain. Sometimes it doesn't rain, or it's not going to rain where they are. So then we look at metaproteomics. These are all the proteins to show, okay, something actually happened, right? We had a protein that was produced. Now, we can arguably say that it had to happen because if a cell is making a protein and it doesn't use it, that's exhausting. That's a lot of energy, where mRNA is like chunk change. So we can make mRNA. But then to make proteins, you really need to hope it happens. But if you want to be sure, then we can go into metametabolomics, which is where we're looking at the metabolites, the interactions with these proteins.
It's so cool. And the knowledge that we have to even do this is evolving rapidly right now. I remember when I was earning my bachelor's degree in biology, we weren't at the place of doing metatranscriptomics or metametabolomics at that point. Those things have really been evolving over the last 20 some years. To allow us to do this now and to look into the environment and see these things. I will mention another poll that we did through the NASA Astrobiology YouTube channel just yesterday, asking people about the scale of viruses in the environment. There are more viruses than stars in our known universe, an estimated 10 nonillion viruses here on Earth, but they only make up a very small percentage of the total biomass. So we gave the audience some options for possible biomass: 14%, 4%, 0.4%, or 0.04%. The correct answer was 0.4%. So they are a very small amount of the biomass, even though there is so many of them. And thanks to things like the work you're doing with, not just the metagenomics, but also transcriptomics and metabolomics, kind of understanding what they're producing in the environment around them, what the bacteria are producing in the environment around them, and how the viruses are involved in that. From all of this, what do you think is the most misunderstood thing about viruses and their behavior, maybe especially in the environment?
It's hard to pin it down to one thing. So if we talk about the general public, I think most people think that viruses aren't really interacting with the environment, that they are moving through the environment. This environment's just a medium. There's no interactions, and they're making their way to a human to hurt a human. And then if we think about scientist level, it's more of Viruses are really hard, right? We're talking about femtograms of DNA. We're talking about particles that are on a nanometer scale, right? So really hard to get at. And science is already hard. And we critique ourselves so hard. I mean, the peer review process is strong, and that's exactly what we want. So we tend not to go for the hardest thing possible, even though that's kind of what I did. So I think there's a misunderstanding is that we can just ignore the viruses and we can describe our system, and that's fine. That it's can be described without the viruses, and that's just not true.
Interesting. Yeah, I feel like, from my own understanding, I really haven't thought that much about viruses in the environment, especially things you mentioned like bacteriophages are the most abundant of the viruses out there. I had another poll. I asked people in my Twitter account what were the most abundant viruses, and luckily most of the respondents, 75%, got it right and said bacteriophages. I want to present kind of a sci-fi idea to you and tell me if it's just bogus and silly or not. Because you've done work in studying viruses in permafrost and polar regions. There was a science fiction horror film some years back called The Thaw that presented this idea that maybe during current issues with global climate change and global warming, with the glaciers receding and permafrost thawing, there could be organisms that have been isolated for thousands of years that could harm humans or other creatures. Is it possible for there to be like old viruses that infected humans long ago that could be buried in permafrost and could still infect us in the future?
Yeah, that's actually a question I get a lot, especially with the recent pandemic. So this is tricky, right? We only know what we know. But what I will say is viruses tend to be where the host is, right? So when humans are sick, viruses around humans, right? That's what we look for. So if you want to look for a human virus, you go to a human that's sick, you'll find that virus. So with permafrost, just to give some context on permafrost, I appreciate you using the word thaw and not the word melt, because it's like a chicken. When you pull it out of the freezer, the chicken doesn't melt. It doesn't change, right? The ice within it melts, right? But the chicken itself is thawing. So we have the chicken there. So permafrost is like that. We'll look at it as frozen ground, and it can have ice within it. For a virus to be there for a human, that means humans would have to be there as well. So if there were humans that were sick with viruses long ago, and they got entrapped in the permafrost, then there could be human viruses. The problem is the viruses need to replicate, and each virus itself it's different how persistent it can be, how long it can survive, how long before the capsid gets degraded, right? So within a human, that could probably last a little bit longer than the environment. If we think about putting a human virus in the environment, that's when it gets tricky. Then you're in an unknown habitat. This is like us on the moon, right? So it's going to change. What I would say is that we for sure will find bacteriophage because there's bacteria in permafrost. We find giant viruses that infect amoeba in permafrost because the amoeba used to be there thousands of years ago. We actually wrote a review about this that came out last year talking about the potential for permafrost to be a reservoir for pathogens. And, that's a great question because for other pathogens, it can be. So if we think of bacteria like anthrax, itself, it's a normal soil bacterium. When it becomes a problem is if it's like in an animal carcass that gets entrapped in the permafrost, right? And when that thaws, that's something that we have to worry about. If we're going to worry about human viruses, we would need to worry about maybe people who were infected with a virus if they were buried and their body was preserved and you came around the body. So just to protect yourself, if you're ever in an icy area and you come across the body, don't touch it, don't go near it. But I'm not aware of a case where somebody's got sick from a virus from permafrost.
That's good to know. So maybe I shouldn't lose sleep over it just yet. So, all of this is fantastic. And you mentioned, like could viruses infect us on the moon, and we talked earlier about whether or not we find a virus on Mars. Is that a sign of life? You just led this paper called Astrobiology, How Viruses Enhance Our Understanding of Life in the Universe. It was published in the International Journal of Astrobiology. For those who want to read more about viruses and this new realm of astrovirology, I highly recommend that paper. It feels like a really great way to kind of introduce viruses as being important to astrobiology and our sheer understanding of what life is and how life might function. Can you kind of give our audience basically an overview of what you mean when you say astrovirology?
Yeah, so first I'm going to give some context. If you want to access a paper, go ahead. I always try to publish in open access, which means that you don't have to pay for it, right? We do this work for government tax dollars. We want to make sure you can be able to read it so we can disseminate the science throughout the public. Now, when it comes to astrobiology, it's kind of a catchy term. Really, understanding astrobiology is a study of life in the universe. And Earth is part of life in the universe. So everyone who studies life on Earth is an astrobiologist. We're asking the same questions, right? Whether you're focused just on Earth, the potential for going into space and the space environment, whether it's on another celestial body, you're asking similar questions: persistence, interactions, survivability, adaptation, right? These are all things we're thinking about. So when I say astrovirology, it's really to put the emphasis on viruses because they've been largely unexplored, especially in our understanding of wanting to go into space, but they're critical. So in the paper, and this was a symphony paper, right? I was the conductor, and we had so many scientists that helped out and different experts in different areas. So I am by all means not an expert in every individual area, so I'll answer questions the best that I can. But we really thought about, okay, well, how do viruses change life on Earth, and how do they interact with life on Earth? What about for people going to space? Because now we're getting to the point where anyone can go into space. So what's happening there? Then if we think about long-term space travel, how do we make sure we have recycling food, our waste, our water? Viruses infect everything, so we need to think about them. And I just want to highlight this to give everyone a better understanding of viruses. Because when I'm saying viruses, you probably are thinking of a cold you had once, or if you had COVID or something else, and how viruses can be so different. There's one you can get sick with and be sick for the rest of your life, or it reemerge later, like with shingles and chickenpox. And then there's other viruses where you can just have a cold and you're fine. So viruses, even though they're small, they're mighty and complex. And we think of three iconic ways in which they can really make an impact. And this is all stuff I'm stealing from my Breitbart's paper. I love it. She knows this. I've told her, and I'll just keep sharing it. So in the first form, right, we have viruses. It's this grim reaper. This is because they're lysing their host. Whether you're multicellular and they're killing cells, or you're single cellular like a bacterium or archaea, they're lysing you. They're killing you, right? And this is releasing your nutrients into the environment. So they're stopping your metabolism. That's impacting the environment. They're releasing your juicy innards into the environment, feeding other organisms, right? And that's impacting the environment as well. When they do this, they promote diversity because they like to attack the one that's winning, the most abundant organism. So by doing that, they're sharing the wealth. They're kind of the Robin Hood. They're sharing the wealth with everybody else and keeping this diversity. So viruses are great for diversity. Their second is as a drug dealer, where when they infect hosts, they share genes, right? So transduction is the mode in which viruses infect a cell, take some DNA, infect a new cell, give some DNA. So they're moving genes around. They're giving genetic novelty out there, and they're spreading the wealth. And the final form is kind of as a wizard. So when a virus infects an organism, and this is when we get into saying if our virus is alive or not, right? So a virocell is this concept where when the virus infects the host, the virus is the one taking over, is the host now, right? So if we talk about that biomass question, it becomes a little controversial if we start counting that as viral biomass, but we won't have to get into that. But as the wizard, right, when the virus is infecting the host, it's wanting the host to do its bidding. So the host is no longer putting out this metabolic output. It's no longer it's no longer respiring, releasing CO2, or photosynthesizing, releasing oxygen, unless the virus wants it to. Some viruses just say, hey, make my progeny viruses, and then you'll explode and die. Other viruses continue to control the metabolism. They can make it more efficient, make it more fit. There's so many different things that viruses can do. So they have a large impact on our Earth and on the evolution of life.
It's really cool to think of them in these different ways as well. I wonder, for my own edification, to my knowledge, we don't really know when viruses started. Were they here with life since the beginning, or did they evolve 2 billion years ago or 500 million years ago? Do we have any way of knowing yet when viruses joined the living processes of life here on Earth?
That's extremely hard. So thanks for throwing the hardest question out there. We have evidence that viruses are still emerging now, right? So If we think of the Earth, geologic time, so 4.5 to 4.6 billion years ago, and we think life arose around 3.8 billion years ago, give or take with our knowledge. There's many different hypotheses out there. If you think of the virus first hypothesis, this is kind of from simple to extravagant kind of hypothesis, where at first we have, oh, maybe it's some RNA, and then we have some DNA, and then we have some amino acids that form around that. Well, that's technically a virus right there. And then if you go up and evolve from that, right? So there's one hypothesis there. There's other ones that make it hard to identify whether viruses were there or not. And I think the reason is we don't have like this LUCA, this last common ancestor for viruses. And it's because viruses are in different groups. We have our RNA viruses, which a lot of them share a specific gene, some don't. Our DNA viruses, you can have this group that shares a gene because it's, if you think about their shape, they're like, oh, this part of my head, my capsid, we share this gene, we're clustered together. But we don't have a universal marker gene. So it's really hard to go back in time with our molecular clock and really see when viruses emerged. We have evidence of ancient viruses. There's work, especially in the oceans, showing that maybe there was that missing link from the RNA to DNA world. But then there's evidence showing with eukaryotes, we have viruses emerging and coming from eukaryotes. So it's a tricky question. And to answer it, we really need to look more. We need to look further. For me, I would love to sample the deep oceans and the deep sediments and in the deep earth to really see if we can have these ancient preserved viruses to try to get at that last universal common ancestor for viruses.
Wow, that's such a cool answer. And I can say we have some questions coming in from our audience already on YouTube. Thanks to those who are asking. I promise I'll get to your questions very soon. I know that we have one kind of similar to that already. Before we get there, I do love just sharing a little bit of what makes us all human as scientists and engineers and researchers as well. You mentioned to us that trail running is one thing that you're really into. I'm wondering, so why trail running? What does that do for you to help you kind of get yourself into a better place? How is that kind of part of your life?
Yeah, so I am somebody that wears my emotions on my sleeve, right? I really care what other people think. And it's because we're humans, right? I value other people and I want them to value me. So running is just a great way for me to get out. It's my time. I'm forced to just focus on me. And sometimes it's boring, right? Sometimes if I'm having a rough day, I am bored and it's not going to be that long of a run. But it helps me focus on me. It helps me get this energy out. And I love exploring and seeing new things, right? When you are in a treadmill, you're stuck in one place. But when you go out, there could be something different. You could run into a snake. You could run into a super bloom, right? You never know what you're going to see when you get out there. And that's exciting.
I love that. Yeah. And I had to say, for me, I love just going for a walk and just speaking to myself and telling myself the things I want to think about. I even use Otter AI to download it and transcribe it for me. It's a great way to get exercise and do some science at the same time. Okay, we are going to do just really quick our faster than light segment. This is where I ask some very simple questions for short answers from you. Our audience loves to hear it. I love to hear it. So let's get going with those. What is your favorite answer to Fermi's question, where are they?
I would say we don't know, we don't know. We haven't explored enough, and we are looking based on what we know and not what we don't know. So we have no idea what we're necessarily looking for.
Oh, I like that. And so, I'm a huge science fiction nerd. I brought up the thaw earlier. Even you speaking about, some of these things viruses do made me think of The Thing, and some of these other stories I love. What stories have inspired you to want to know more about life in the universe?
Yeah. So biology is hard and just exciting. It's not like other sciences where you see something, you'll see it. We take technical biological replicates, we still get some different answers. We have to do so many studies, and in this case, it'll do this, in this case, it does that. So I think that stories that excite me is when there's brand new research that comes out that just makes everyone go gasp, oh, and then you can literally find out 5 minutes later, just kidding, that was not a thing, or it was an N of 2, or we found it doesn't always happen, right? So that's kind of exciting. I also get really excited when people are first learning and you see that spark where something excites them. And in their head, for at least 5 moments, that's what they're doing. That's what they're going to be doing. They're sure of it, right? So that I feed off that. That's exciting to see and to see people passionate about stuff. Um, for science in general, though, I wasn't a kid who grew up thinking, oh, I love science. Science is amazing. Science was a course that I just kind of got through. Um, it was really in college. I had this strong passion for climate change, right? So I kind of thought about it from the political side, but then I didn't want to just be a politician talking about science that I don't know or arguing against science I don't know. So then I took some science courses, and it's really looking back in time and seeing these things people discovered that a lot of times it's by accident or it's a lot of hard work. We give credit to a single person, but there's been so much research leading up to that. So in my lifetime, even if I don't win a Nobel Prize, the research I've done, other people are building off it. We're a community. So I know I'm helping in some way. Sorry, that was a lot of different answers.
Yeah, no worries. I agree with that spark that you see in people's eyes when they learn something new, and it's this fun discussion, this conversation that we have where we learn something together, and that's part of the human endeavor of science and learning. If you could go back to the beginning of your career and give yourself some advice, what would you say?
Encase yourself in a suit of armor. Life is hard, and going through college. So when I say college, this can be anything. Trade schools included. A university is just a collection of colleges. You learn things, right? And people offer their opinions for you to learn, and then you have to learn what you're told to learn. And it may be a little bit different depending on where you are. But then you go into grad school, and it gets even harder because you're not only supposed to learn, but then you're supposed to add now. You have to contribute to science. And to make science valid, fair, and equitable, we have to make sure that we're arguing with each other. There's a peer review process that science needs to go through. So we get attacked all the time. And it kind of makes us hard. And really getting back in tune, finding ourselves, making sure we have hobbies, making sure we can relate to people still, and not become hard. I would say for a lot of people, graduate school is a really hard time, myself included. So I would, if I could just go back, I know I'd trust myself and say, hey, you're going to get through this. You can do it. It's all about perseverance, persistence, right? You don't have to be the best person out there. You just keep trying.
That's fantastic. I myself, I think a lot right now where AI is at right now with all this generative AI going on, like ChatGPT and Bard, and we just had this recent test of Starship from SpaceX. There's so much going on right now in our world with science and technology. Things are progressing very quickly. And I'd say right now, if you go through any social media site, there's, kind of some pessimism right now about what the future might hold for our species. But it also feels like there should be a lot for us to be excited about. So I wonder from you, what's something that excites you about the future?
Yeah, this is hard as you get older. When you're a kid, everything excites you, right? We have our creativity, our imagination is huge. I think talking with older generations can be helpful. Like a lot of times we'll brush things off, but if you really listen, you'll see the problems we have now are problems we've always had. There's always been the pessimism, there's always been the excitement, and you realize there's always just something new. So what excites me is that for science in general, we're moving to a lot of interdisciplinary science where we're working together. I no longer have to be an expert on this one gene that does this. This person no longer has to do that, right? We can start to understand these fields and come together, just like the paper we published, where we're all experts in different areas. We were able to unite on a topic that we agree on. We could argue, we could debate, and we have something to show of our works of working together. And I think that's something we need to really think about and excites me for the future is the idea that science is moving in a way where we're uniting as a science. We're seeing more and more proposals and calls for us coming together, not just this person can do this, but really looking at a system, right? And that's been the crux of my research since the beginning. I've always wanted to look at the system. So the emerging idea of system science is good science, I guess.
Very cool. Now you work now at the Lawrence Livermore National Laboratory. I think some people aren't aware that there's like these different things you can do throughout science. You don't have to just become a professor and work at a university. There's also research institutions. There's various ways to work throughout various industries that are related to science and engineering, as well as national laboratories. So what would you say is the best part of your job?
Okay, so we're never going to just say it with one thing, right? What I love about a national lab, and they're kind of this black box that doesn't get advertised that well. You grow up going through academia thinking, oh, you need to become this professor. Or if you go in industry, that's where the big bucks are. I would say the national labs are a great medium in between where we publish. You can publish as much as you want. You don't need to publish if you don't want to. It's really what kind of science you want to do. What I do like about the national labs is that our government, Congress, sets aside money to make sure that we're on the forefront of science, right? So we're really always pushing the envelope. And then we want to work with other people, right? We have so many calls if you go through DOE's website to really get people, universities, come and work with us, whether you're community college, undergrad, high school, really working with the national labs. I was worried when I was going to come to a national lab. I'm like, oh, I'm never going to be able to teach. I'm never going to be able to connect with students. But that's not true. We have so many universities nearby, and even in other states, I connect with Georgetown quite often, where I can mentor students and have no issues with that. And if I have to say my favorite thing working at the lab is the freedom I have to be the scientist I want to be. So I'm not going to speak for a professor at a university, but you generate this lab, you're expected to do a certain thing. That's the science you kind of go down. You can kind of shift here and there, but I don't know if you have as much freedom as you do out in a national lab. I came in, I was a virus guy. I'm working with another group as a virus guy. I love it. But there's other projects I can work on that may not even involve viruses, or I can actually introduce viruses too. And it's a system maybe I've never worked with. And I absolutely love that it's just so open and welcoming that I can just work with other people here at the lab.
And that's excellent. Yeah, I think it's one pathway people don't think about that much. We will answer it up here as soon as we can to the audience questions. I promise for those asking questions right now in the chat, I will get to them. But first, my favorite of all of our Faster Than Light segment is this question. What is an unbelievable science fact that still blows your mind?
Oof. That's really hard. Yep. I would really, maybe it's a culmination of facts. It's the idea that there's things all around us that can affect us that we can't see, right? We think that we see something and that's that, but our vision is actually pretty bad compared to animals out there. So I guess, okay, let's think about this. I think a fact that I really like is growing up, you think of humans rule the world. It's all about humans. You're not thinking about animals and everything else. But we have literally been on the planet for seconds and minutes, right? Geologic history is billions of years, and we're just this little blip that's on here. So the Earth had been around before us. The Earth will continue to be around without us. So we are just renting this hotel. We're renting our time here. And so it's just crazy that there was just this whole Earth before humans.
I like that. Yeah, there is so much time that we weren't here. we are just a blink of an eye, and who knows what the future holds as well. I will open it to our audience Q&A now. We'll start off with a question from Twitter from Dr. Jim Pass. Jim asks, what risks may occur to humans settling on Mars over time, assuming that viruses or living extremophile bacteria exist there, especially for astrobiologists. And I think I'll add to that, do we have any knowledge of how viruses change in the space environment yet?
Yeah, okay, so I'm not a human virologist, right? But I do feel I can answer this. When we send people into space, your microbiome does change. And we think it's largely due to stress. It's similar to the stress we see here on Earth, right? We have microgravity, there's just a whole slew of things going on. And if we're going to be on Mars, it's going to be an environment different than Earth. So we're taking Earth organisms like our bacteria, our archaea, our fungi, and biological entities like our viruses, to a habitat that they're not used to being. So even though they're within us, we are keeping homeostasis to our environment, and they have to adjust to that as well. So there are risks we have to think about. They're going to act different. And if they act differently, is that going to throw our system out of whack? We need to think about our health. We also need to think about if it's going to change our mental state, right? Our gut microbiome, including viruses, really affects our mental health. So there's a lot of testing that we have to do. And we are doing this now. There's people on the International Space Station. We see that when we send people to space and they come back, their health does change. If you have something like herpes or chickenpox, these are latent viruses, they come back, they reactivate. These people are not happy. You get shingles. And this is something we have to think about when we're sending people into space. Now, it doesn't always happen, but there's definitely research we need to do more there. So I would be worried not of a crazy Martian virus hurting us, right? We're going to be foreign. Why would they want to infect us? Viruses are usually host specific, so they're not going to care about us. But we need to think about our health and how our health is going to change. I think we have this idea that, oh, as long as we have oxygen, we'll be good. No. We have a whole system that we need to think about, and we really need to ask them what they want.
Indeed. We have a question from Kelly Yoak on YouTube. The question says, does a virus attack fat or protein? I'm thinking what their question is how does a virus get into a cell? What is the pathway for it to actually approach and attack the cell?
Yeah, I like your interpretation of it. I'm not sure I know how to answer that. So you can have viruses that can have tails, like a common bacteriophage will have a tail. It attaches to the host. There's receptors that bind. It opens up, injects its nucleic acid in. Or you can have enveloped viruses where they have their fat around them. They attach to the host cell membrane. They fuse with it, and the whole capsid comes in. So is that what you're asking, I guess?
I think that's what Kelly was going for. Yeah, I think so. Here's a question. I'm not sure how you feel about this one. This is from Rendering Reality 3D Animations on YouTube. Can we discuss the similarities with cryopegs and ice brine channels as having possible root connections to the origins of life? For example, for the formation of the first capsid. I'm guessing that they're, given your knowledge of Arctic and polar regions, they're curious of your take on things like cryopegs, ice lenses, and ice brine channels for being potentially important for the origins of life.
Okay, so caveat here, I've studied permafrost, specifically the active layer above the permafrost that seasonally thaws. Some colleagues I worked with, like Dr. Pacifica Summers, has done some cryopeg work. They would be more astute to be able to answer these kind of questions. But what I will say is, if you're looking for early life, like what I was talking about earlier, going to the bottom of the deep ocean below the sediment or deep earth, we need to get to an area where life can be preserved and not affected. And the problem is, we have, with plate tectonics, we have the earth is moving. The earth is constantly changing. Things are being destroyed, being made. So we need to make sure it's somehow preserved in a way where it's not going to get in a subduction zone or be lost, or it's not going to just be, okay, so let me step back for a second. If we think about fossils, most fossils are found in the ocean, right? They can settle, they can get preserved, it's less likely to get disturbed. Or on land, if something settles, it's going to be kicked around, there's going to be climate, weather affecting it, right? So I think the idea of a cryopeg is that it's this preserved sub-ecosystem. And that's definitely something that we should be exploring more. I would also argue that we should explore our ice cores from our glaciers, and not just the glaciers in the Arctic or in the Antarctic, but our high altitude ice cores that we have, and really start looking for viruses in there to look at past viruses. And I think a common issue we're going to have, and this is where we really need to push the envelope here, is how do we work with low biomass, right? Viruses, there's not that much there. I'm commonly from soils having to pull nanograms to picograms of nucleic acid to look at them. So we need to become more sophisticated.
Very cool. I agree entirely. User AC on YouTube asked us a question about whether or not a chemoautotrophic organism could evolve in rocky exoplanets out there, much like they have on Earth, and having anaerobic metabolisms that aren't dependent on photosynthesis, but only on minerals for energy. So first off, AC, that's exactly what we know of for life here on Earth, and so it makes entire sense that it could happen elsewhere. And so I think for Dr. Trubl, let's explore this a little bit more, though. What do we expect for possible viral populations out there in the universe? For instance, if we find a world where photosynthesis never evolved, do you think that would change the kinds of viral populations we might see that are out there?
Yes and no. I mean, there's many viruses that don't infect photosynthetic organisms, right? So they would be arguably the same. I mean, I think life's going to be way different elsewhere we find it, right? But we don't have something to compare to, so we don't know. But there's viruses that infect organisms that don't photosynthesize. It's not like it's required for a virus. A virus just needs something that's going to have metabolism, it's going to generate ATP to take advantage of. That's all it needs. So whatever weird biology is out there, the virus is going to fit to it no problem. I think the question we should be asking is when we're looking for life detection, we need to think about the virus influence, right? So for biosignatures, a virion itself is a biosignature. If we're thinking about places where we can't land rovers and we're just wanting to sample atmosphere or maybe measure from a distance, we need to think about how viruses are influencing that signal, right? So we're thinking about stable isotopes and gas products for CO2, the fractionation of when it's made. If a virus is infecting a host, it's using the genes differently. It's pushing this machinery. So the fractionations can be different. So the isotopic signature of the gas is going to be different. So just to give a background, when we're looking at a gas like methane, we look at the fractionation to really say, hey, did it come from life? Did it not come from life? We can even get down to what exact metabolism did it. So for methanogenesis, it can be hydroneotrophic or cytoclastic. You can have, if I'm going to say this right, hydroneotrophic is -80 per mole, just to throw random numbers. Cytoclastic's around -60. And then you can have variation, right? So life, there's always variation. What variation is caused by the virus? What if it makes something look more abiotic because it's just pushing the genes? And really this fractionation comes from, as life, when we use things, we want to use the lighter isotope version. Sorry if I'm getting too sciencey here. And when the virus takes over, the virus can just put the pedal to the metal, right? It's like, let's go, let's get this done, right? So that selection is not going to be, it's not going to be happening as much, right? And so a lot of this is more research needs to be done, right? So I'm maybe speaking too much on this. But when we're looking at life elsewhere, we need to think about what are viruses doing? And we need to be thinking about on Earth to better equip ourselves for looking for life elsewhere.
I like that a lot. And for our audience who maybe don't quite understand this idea of fractionation of stable isotopes, it is worth looking into. There's some really great explainers out there. What Dr. Trubl is saying, basically, when it comes to life and enzymes that life uses to have metabolism and do things, life tends to prefer using the lighter isotope over the heavy. There are some very good chemical reasons for that. And so we see a fractionation or a change in the isotopes that are available being produced by life versus non-life processes. And so that is very intriguing for us. Now, I will say our next question, maybe someone just could use some help in the lab, it sounds like. So Dan in the cave has asked, they've just started their PhD at the OU in the UK. Investigating microbial communities in the Botswana salt pans as an analog for Mars. And they want to know if you have any tips for how to separate viral DNA from their soil core samples.
Super cool. So that's always great, especially when you're getting to do research. When something is sexy, cool, it makes it easier to talk about. And as scientists, sometimes it's hard, right? Like if I have to talk about something in poo, you're like, well, this is poo. Or if I have to talk about this and it's not that exciting. So having something like that, that's great. That's always enjoyable. Yeah, so I can help you more if you want to email me. We can chat about it. But what's really hard is you need to be thinking about what is binding to the virus. And is it causing the virus to precipitate out? Or is it is it protecting the virus? So you have to be able to tailor your methods to your ecosystem, right? So you have to think about the virus interacting with what's going on with around them to really be able to resuspend and pull the viruses out. This is a major challenge we had in soils, especially the peatland soils that I worked in, because there's a lot of organics. So in peatland, especially in northern latitudes, you'll have plant biomass that just accumulates on the surface and it's slowly degraded. So it's very fluffy. You kind of walk across it like a Tempur-Pedic mattress. It's really nice. It's bouncy. It was cool to observe, right? You can also have certain plants, like mosses, like sphagnum mosses, like bogs that are in bogs, sorry, that produce certain acids that inhibit growth, right? So you have a lot of this plant biomass going on. Now, the trick was to get the virions, the virus particles, off the soil particles and this plant biomass and make it pure to be able to resuspend that. And that's difficult. So we'd do a lot of testing in different buffers. We'd do a lot of physical desorption methods to get those virions off. Another thing that's helpful is by doing size filtration. So viruses are really small. And this, again, you're going to be pushing out most viruses. You have to think about what viruses you're critically looking at. So if it's double-stranded DNA bacteriophage, you can run them through a size filtration, like 0.22 micron filter, and really isolate your virus. So I don't want to go too much more into it. I'd love to be able to have a conversation. So please just email me. I'm happy to talk with you.
Awesome. Yeah, and for anyone starting a PhD program or any realm of research, always reach out to the people who've done the work already and can help you in the laboratory. There's no reason to build new methods or bang your head against the wall. I learned that the hard way myself as a graduate student when I spent way too much time often in the lab trying to figure out how to do something when all I really had to do was ask somebody else who'd done it already. It would have saved me a lot of time. So definitely please reach out to the experts when you have questions on how to do your research. Our next question, I think I'll take myself. Marvel Sushante on YouTube has asked if we could explain how coxolithophores are important to astrobiology. So for maybe our viewers not really aware, a coxolithophore is a marine phytoplankton. They're a very prevalent calcifying organism on this planet. That means they take calcium and the carbon dioxide that turns into bicarbonate out of the ocean and make calcium carbonate. They've been involved in forming rock layers of calcium carbonate. When you think of the White Cliffs of Dover in the UK, those cliffs of Dover are chalk that are formed from the bodies of organisms that have calcified in the ocean from long ago. And so it's important for us in astrobiology, if we're trying to understand the nature of life, how did life originate here, or did it originate elsewhere and come here? How did it evolve and change with our biosphere and our world over time? And how can we find it out there? And so understanding how organisms calcify in the environment, how they create mineral structures in the environment is important for understanding what life does and for what we could look for out there. Maybe there have been calcifying organisms on Mars in the past or ancient Venus long ago. Would any of those signs be left for us to find now? We truly don't know yet. Our next question comes to us. Aruna Devi has asked, can we find viral fossils in archaeological remains?
Yeah, that's again hard. So, when we're looking for these fossils and we're looking for ancient preserved life, right, we're looking at stromatolites, and we have great evidence of stromatolites all over the world. I was fortunate enough to do the Aragon geobiology course in 2014 and got to go out and actually sample some stromatolites myself. That was invaluable. Those are great experiences. So, when we're looking for these fossilized organisms or for biological entities, what's really hard is discerning between, is it abiotic or is it biotic, right? And that's tricky. We can't just extract nucleic acid out. So we possibly could find a virus. It'd have to be an iconic shape, like a bacteriophage, and there'd have to be great evidence to show there was no abiotic process that could help form that. I don't doubt that it's out there, but I think it's easier to find with the larger organisms just because of where our resolution is with our current technology. Not to say it's not possible.
Well, let's talk about what's possible for just a moment then. This next question I really appreciate from J.A. Holt on YouTube. To what extent are we sure that life originated on Earth? We aren't. Might life itself have been brought here? Now Jay says from another universe, and then the question goes to viruses. Now, we wouldn't say another universe, Jay. We don't have any knowledge of another universe. The very idea of a multiverse is really rooted in some mathematics and and some speculation, but we have no evidence that multiverses exist yet, let alone that life could be brought here from that far away, wherever that is. But there is this idea called panspermia, that life can be moved from one world to another, and that maybe we are Martians, or maybe we're Venusians. Maybe life started on another world and then came here. And so I think, Dr. Trubl, for you as well, we could have some fun and speculate here. Could viruses have come from another world and then been brought here? What do you think?
Absolutely. I mean, that's viruses' role in life. They don't have flagella. They're not moving around like that. They're hitchhikers. They like to get in their host, move on over to a different host. Sometimes they survive outside the host. Sometimes they go in the host. So I think they're adapted to survive. So that's possible. Yeah.
Love it. So we have another question that I also really appreciate. I'm glad our audience is like thinking speculatively here about what's possible with viruses and life. So AC has another question. They've asked, what makes any of us think that viruses could be found in other exoplanetary ecosystems and not just be a fluke of terrestrial evolution? And that's a good question. Do we have any reason to assume that viruses will exist on other worlds?
I mean, we could say the same thing about any life. Um, so if we think of life starting off as viruses, if we're looking for life elsewhere, then there would be a good chance that something of a virus would be there. I think there is actually a better chance of a virus being found than some of the other things that we are possibly looking for. And the reason is because all the different things viruses can do, right? They move things around, right? They're these mobile genetic elements, and they're critical to life. So if we're going to find life elsewhere, we're undoubtedly going to find viruses or a virus-like entity.
Very cool. All right, so I think we have time for one more question. So user Florian Boreas on YouTube has been asking about how viruses might be able to have more resistance to the space environment, to other environments out there, how they could survive and develop resistance to extreme environments in the cosmos. And, I think, we kind of have to think about what viruses are. They themselves are not the ones that are producing necessarily the resistance. That they're taking over these populations of other cells of other beings that have developed resistance. So I think maybe a more enjoyable question for us to finish off here, Dr. Trubl, would be what is our knowledge right now of how viruses impact extremophilic organisms, and do they have any kind of role to play in the diversification of life to fill these more extreme environments?
Yeah, absolutely. Man, there's a lot of good research out there. For anyone who's thinking about viruses, especially in extreme environments, I would tell you to look up Dr. Kenneth Stedman, professor at Portland State University. He was a person that I really reached out to. He's been a great mentor to really understand how viruses are really involved in astrobiology in extreme environments. He has a slew of great papers. So, viruses, it's what we say is an extreme environment. It depends on the host, right? If a host is living there, is it really extreme? That may be where they thrive. It may be if you put them in something we find as a good environment, that they actually get outcompeted. They're not happy. So when we say soil's harsh, I say not. I think it's harsh to us, right? So I will say extreme to us, but not necessarily to that organism. We know that viruses will take genes from their hosts. We're still trying to figure out exactly how everything happens and why it happens, but they can have these genes that we'll call auxiliary metabolic genes. And sometimes when they infect a new host, another family member within the host, they can take over the host and cause the host's genome or metabolism to express that gene and make the host more fit. So viruses can definitely help hosts survive, and not only survive, but maybe become more fit and outcompete others in the environment.
Very cool. Great way to finish off our episode. Dr. Trubl, thank you so much for joining us for Ask an Astrobiologist.
Thank you.
So if our audience did want to learn more about your research or your work, what's the best way for them to reach out and find you?
So I really like Twitter because it's very informal. I just messaged other scientists and they get back to me. And so I've come across, not calling it Twitter, but science Twitter specifically. All the other shenanigans that go on, not really something I'm concerned with. But just as a community of scientists, it's a great way to reach out. You can also feel free to email me. Yeah, any way that you think you can normally email someone, I'm happy. I'll check my emails. I think what's hard is sometimes people will message me just on LinkedIn or something. I don't always check everything. I probably check Twitter the most. But a good old email. I check emails every day. So let's just go with email works best.
Love it. All right, Gary, thank you so much for joining us. It's been a great time having you on the show to talk a little bit about astrobiology. There's still so much more for us to explore and to learn. For our audience who are tuning in, if you want to follow along with more going on for Ask an Astrobiologist, of course, we have the hashtag on Twitter, hashtag Ask Astrobio. You can also sign up for the mailing list for the NASA Astrobiology program. You can learn more about current events, new opportunities, and things that are going on across the realm of the NASA Astrobiology program. And so to all of you for tuning in, thank you so much for joining us. And as always, until next time, stay curious.